My name is McIntosh. I'm the director of the Institute for Neuroscience and Neurotechnology at SFU. Before I introduce our special guest, I'm just going to mention there'll be another in-seminar on the 11th of April, I believe it is. It's 25th, one of the two. April. Stephanie Willard from UVic will be talking. She does work in neural tissue engineering, which includes building scaffolding as well as organoids for regeneration, as well as for doing cool things with 3D printing.
That's awesome.
So I'm happy today that Logan Grosenick has joined us. He's visiting from Cornell. He's an assistant prof from Cornell, the medical school, specializing in methods in the interface of machine learning and psychiatry. He does very interesting work on subtyping, and that's what we're going to talk about today.
That's right.
Logan, please.
Thank you very much, and thank you for having me. It's great to be here. Today I'm going to talk about one part of the work in my lab is really focused more on the sort of human scale, like whole brain. Can we develop what we're going to call biological subtypes? I'll tag that in a second. For psychiatry, and I'm going to kind of give you an overview of sort of a little bit of their origin story, some issues with them, and sort of where we are now. So what is a biological subtype? What we mean is kind of using, and I'll get into the details further, but we want to use neuroimaging or neurophysiology data alongside psychiatric symptomology to define different groups of patients. And the key here is that these different groups of patients, these subtypes, will predict treatment outcomes, right? Because we already have a lot of ways of clustering things, but what hasn't happened historically using just data based on clustering symptoms alone is coming up with categories that actually predict treatment outcomes. So I'll just introduce initially the problem here. The Diagnostic and Statistical Manual of Mental Disorders, the DSM-5, is in many ways a great achievement, but in other ways a catastrophic failure. So the former head of the NIMH, Thomas Sinzel, described a lot of the methods that are in here as having 100% reliability and 0% validity. So they're very reproducible, and they tell us nothing useful for actually predicting treatment outcomes, or not as much as we would like, certainly. And at the root of that is a troubling heterogeneity. So I'm going to talk about depression in some detail. Depression in particular, if you get 5 out of 9 items in a checklist, you're depressed. And so there's combinatorially many ways, different combinations of mood, appetite disorders, lethargy, that can lead you to the same diagnosis. So you naturally have some patients who have very high anxiety and bouncing off the walls, and others that can't get out of bed with inertia and psychomotor retardation, and they all get the same diagnosis. So if you're a biologist looking at this, it's very natural to think there might be different biology underlying different parts of this diagnosis. And so that led us to a pretty simple, a very simple hypothesis for a biologist, although historically this was not in psychiatry, the way that things were done, and that is that we would like to parse diagnostic heterogeneity using neurophysiology. So the very simple model here is that there's some underlying actual neurobiology that's driving both the clinical symptoms that we're seeing and measuring and the neurophysiology that we're able to measure. And then if we can use those to triangulate, we may be able to use biology and clinical symptoms to start to define biological subtypes. I don't know why this is lagging so badly.
Less oxygen up here in the front of the mountains. Less oxygen.
It probably is, yeah. My computer has altitude sickness. So when I say neurophysiology, I'm going to talk about some measurements that I think we're all very familiar with. These aren't particularly new or fancy, but we're using resting state functional connectivity. In particular, we're going to be using the Power Atlas. This is a very nice, robust, validated parcellation from Jonathan Power, who's my neighbor now at Cornell. So there's 258 different circular ROIs across the brain. And looking at those, we could take a patient's data from the scanner and quite simply look at 2 of these spheres over time, take their correlation. For example, here's the correlation between dorsolateral prefrontal cortex and posterior parietal cortex. That gives us a single number that over the course of that resting state scan summarizes the correlation between those regions. So pretty simple measure overall. Good for patients though, because you can put them in the scanner. You don't need to do particular tasks. So it's been of general utility, although there's plenty of good critiques of it. But even this pretty simple measure gives us a fairly complicated data set. So with 258 ROIs, we end up with on the order of 30,000 correlations above the diagonal here in the correlation matrix. And a lot of these are driven by things that might not be that interesting to us. So there's a lot of things we don't think are of interest to us. Sources of variability that may not be related to things that would be relevant to psychiatric diagnosis or treatment outcomes. So to us, solving this was a very important problem. Again, coming back to this very simple view, we have this very highly multidimensional neurophysiological data that we need to figure out a way to filter through. And we have something, we're going to go with something a little bit richer than the DSM-5. We're going to use the Hamilton depression rating scale for depression, which is a 17-item scale with a number of subscales that map onto different components of depression, including anhedonic measurements, anxiety measurements, measures of sleep, disturbance, and so on. So these are kind of our clinical scales. And again, in keeping with this very simple model and hypothesis, we'll put in resting state functional connectivity as our neurophysiological correlate. And we're going to use a very simple model that imagines that there's some true latent brain state that is producing both our clinical measures and our neuroimaging measures, both of them multivariate. Because they're both multivariate, we're going to use a very old method. This is cutting edge work from the 1930s by Harold Hotelling. We're just simply going to look at maximizing the correlation of some coefficients on the multivariate clinical measures and the multivariate neuroimaging scores. The idea is that this is one way, again, making a lot of assumptions, to get at a underlying latent variable that would be the things that are maybe relevant both to the clinical measures of interest as well as the neurophysiology. So this acts in some sense as a filter on both. Using this very simple model, in this case, a multi-site study. This was collected around 2015, 2016 from a number of labs, using in treatment-resistant depressed patients. And there's a lot of additional details in the paper about homogenizing these data sets and controls. But the punchline is that we can run canonical correlation analysis. This is in the training data, so you can already see it's overfit. But it defines for us sort of two primary significant dimensions from the canonical correlation analysis. So we have these canonical variates that relate to the load mainly onto anhedonia, a little bit onto psychomotor retardation. And one that loads more heavily onto things that look like anxiety and insomnia. And again, just a warning, these are fit to training data. So we'll get to generalization of it a bit later here. Critically, we could then go into the latent space, that two-dimensional latent space in this case. Again, quite simple. Apply hierarchical clustering, very standard, another off-the-shelf method for clustering, using standard methods like Durgan and CH and other scores. They agreed on 4-cluster solution. We checked that this solution was stable by repeatedly subsampling, including the clusters again, and looking at cluster membership. Did individuals fall in the same cluster? They did. And so we convinced ourselves that there are 4 clusters here. Again, we'll come back to whether or not just how split these clusters are later in the talk. But we have 4 clusters, and what was very exciting and really the important part of the paper here is that some of these clusters responded to antidepressant treatments significantly more than others. That's the punchline, right? That's what we care about. The clusters are of interest for a lot of reasons, but we care about, yes.
Are the antidepressants cohorted as well, like different types?
They are. So this I should say this antidepressant treatment is in particular repetitive TMS. This is transcranial magnetic stimulation over dorsomedial prefrontal cortex, and so it was just one treatment, and it was standardized in this case.
Great, thank you.
So that was also another exciting component of the paper this one. But for the sake of our biotype story here, the important thing is that there's both a dimension and clusters along that dimension that responded to treatments and ones that did not. Okay, so just to quick summarize this first study. So we've identified these two brain behavior dimensions using just canonical correlation analysis. And I didn't go to this, what we did prior to that. CCA has some stability issues. We did some feature selection, there's some details. And this predicts individual differences in anxiety-related symptoms. If we cluster in this space, we get 4 putative subtypes of depression. Furthermore, the subtypes are associated with different clinical symptom profiles. So I showed you that, and they also have different connectivity phenotypes that we can develop biomarkers from. I'm not going into those details. I would recommend looking at the paper for that. We currently have a large-scale clinical trial underway in about 400 patients following the efficacy of the biomarkers we developed on these subtypes and their ability to predict treatment outcome, new treatment assignment, and advancement. So this was very exciting. It provided the first indication to us that this biological subtyping might get us past like where we've previously been in psychiatry, where seasonal depression, atypical depression, bipolar depression, these things you can cluster and find them symptoms, but they don't track, they don't predict treatment outcomes, and they don't give you good tools for treatment assignment. So this was very exciting. Given this, we wanted to see if this was a general trait of, or a potentially general method to apply to heterogeneous disorders in psychiatry, since this is a known problem in psychiatry that we have these very big bigs. And a next logical place to look was autism spectrum disorder. Autism spectrum disorder was created in the DSM-5 by amalgamating 3 other groups themselves of dubious heterogeneity. So we knew it to be very heterogeneous. And in this case, it's interesting. We didn't have a treatment candidate, obviously, but there's very strong genetic predisposition to autism, right? There's strong biology associated with autism, and we wanted to see if we could both say, okay, this can predict treatment outcomes. Also in heterogeneous disorders, we can potentially relate this back to biology. So we went, furthermore, there was publicly available data that was also important that we could look at. So the question here, could we use the same approach, again, fairly simple-minded, to parse heterogeneity in autism using neurophysiology? It's a very similar approach, just now we're using the resonant state functional connectivity. We're going to use the ADOS2, which was available to us with these really nice data sets, the Abide1 and the Abide2 data sets, which had, after we corrected for motion and did some other housekeeping, we had about 400 subjects to include. And this is very nice work by postdoc in lab Amanda Booch. Back to, there she is. Really a heroic paper. I'm going to go over just some details of it, but there's quite a bit in there. I think she resubmitted with like 50 supplementary figures or something like that. It's very detailed. So check it out. So improving our methods a little bit, just one methodological detail. We're going to use regularized canonical correlation analysis. In the interim, we discovered this predictably helps stabilize our solutions. The reason for that is you can really think about this problem up here, this old Harold-Hotelling problem, as a generalized eigenvalue problem, where essentially you have, but now we're looking at the cross correlation matrix or cross covariance matrix, so between our X and our Y data here, and we're actually secretly whitening in both the rows and the columns, of this matrix using the sample covariance matrices for X and Y, and that becomes unstable, right? In these large data sets, they're sort of underdetermined systems, so it's very natural and reasonable to, just like you would in ridge regression or Tikhonov regularization or your preferences, just add the stabilizing element to the diagonal of your sample covariance matrix. And this helps stabilize the solutions quite a bit. It helps to generalize that sample more, and also allowed us to include more variables as we did the feature selection, since with just CCA, things are a lot less stable if you have any sort of multi-collinearity in the columns, which most of us do. So another approach, you can also do PCA first, and to stabilize things. This worked better in our hands and allowed us to do feature selection, which has yielded better results. There's some details on this sort of methodology based on that work from the 1970s. It's similar to work that has been done in the 1990s, since CCA and PLS are sort of potato, very similar methods. Okay, so one other just methodological note, and we'll come back to why this is critical as well. I think this is probably known pretty well to everybody in this room, but you really need to do nested cross-validation. And I also just want to make a little PSA. Please, when you do nested cross-validation in your papers, include a diagram like this. It really helps reviewers to go through and see exactly what you did, and Amanda made a really terrific one here. So we're going to take our training data, we're going to keep some test data aside, obviously, for the end, that's our out sample. And then as we optimize all of our parameters, because we have those two lambdas we had to fit for the regularization parameters and the regularized canonical correlation analysis, we're going to fit them on this validation data, come up with our final model, and that will be tested eventually on our test data. We're actually going to repeat this whole process 1,000 times to kind of get the overall estimate on the test data, but that is completely held up each time. All right, some of the details I'm sure I'll skip. So we do this in this BIDE data set using the ADOS2 as our X, say, and the resting state functional connectivity from the same data set as our Y. And what we find is there's actually three significance, and significance here is against an empirical null distribution built by bootstrapping the data. We get a verbal IQ related dimension, a social affect related dimension, and a restricted and repetitive behavior related dimension. This is not especially surprising. There are three measurements on the ADOS2, and they are verbal IQ, social affect, and RRBs. So there's some interesting details too. There's some mixture here, and we'll see why actually this becomes interesting for the clustering in a moment. But this makes a lot of sense. And again, these are three core symptoms for those of you who aren't familiar with autism spectrum disorder. So these are verbal intelligence, social affect and social relations, and restricted and repetitive behaviors. And the light dots are actually on the held out test set, and that's where these numbers are coming from. So we're getting canonical correlations of around 0.2, 0.18, not huge, but significant there with effect sizes for cones deep around 1, 4.7, 4.4. Okay, so again, now we have this sort of dimensional representation of, again, this is a low rank representation of the covariability between this very large resting state functional correlation network and the symptoms that are of interest. And again, what we think this is really doing for us is helping us filter this variability of brain data, which is variant on a lot of different levels for the things that are actually relevant here for the diagnosis. We're just going to use hierarchical. We're doing this two-stage procedure. First, we embed in this lower dimensional space. Then in that lower dimensional space, we apply hierarchical clustering. We once again apply 6 different methods. Here's the CH criterion. They all pick out 4 clusters. So we found 4 clusters. Yeah.
Are these clusters, I forget the technical term for this, are they independent in that group membership can only occur once for each observation, or are they?
They are. So these aren't like probabilistic clusters. Hierarchical, like agglomerative clusters, such as Ward clustering in particular, which is the flavor of that, is going to at the final where you cut the tree is going to be like, this is a 1, this individual is a 2, this individual is a 3. It's not probabilistic.
Okay.
We do again, and there's some supplementary figures on this. Resample the data a lot of times, and then use this Hungarian matching algorithm to just match the classes so that we know which class corresponds to which class, and then we can go through and say like how many times of the 1,000 times we resampled was this person in the same class.
So you generate the probability
exactly
of occurring in the cluster by resampling. Okay.
Exactly, and that's kind of how we assess the stability of the clusters here at the end of the day.
Okay, cool.
And then so what's very interesting is now we can go and look at by subgroup, by these 4 subgroups, do they have distinct clinical symptoms, right? What are those different symptoms? So we have for social affect, there's clearly one subgroup here that is very, very low on social affect. Restricted and repetitive behaviors, we have a group that's so this is quite impaired. These three groups are impaired and this is not. And there's this interesting split number of IQ. So again, there's only 3 dimensions. We end up with 4 clusters, and it seems like that fourth cluster is coming out of a differential on verbal IQ, which would certainly be consistent with what historically would be like Asperger's or something like that, a fairly highly functional, high verbal IQ group. There's also some differences in total severity. So this group is relatively spared. These subjects have distinct patterns of atypical connectivity that are pretty consistent with what we would imagine in the literature. I'm not going to go into all the details here. There are 2 display figures and many supplementary ones on this. The point here is that there's interesting differences that we can pull out in both the behavior, or I should say the clinical scales, and as well as the brain connectivity. This is atypical connectivity relative to a large cohort of healthy controls. Importantly, if we take this model and set it aside and go to a completely different data set and use the exact same, instead this is the NDAR data set, so which has many fewer subjects than the ABIDA after we correct for motion and so on, and availability of the subscale. The NDAR data set has about 85. We apply the exact same protocol without it ever seeing the ABIDA data in any way, shape, or form. And we get results that I found to be surprisingly similar. Like I was expecting it to be somewhat worse. This particularly in terms of, again, this is here we're just looking at them, but qualitatively quite similar in terms of the atypical connectivity patterns that we see, as well as the patterns of behavior per subtype. So that was a great sanity check for us. And again, my pointer is more to convince you about the existence of biological subtypes than it is to go into the details of connectivity and behavior for each of these examples. I'm happy to do that offline, and the papers have plenty on it. But that was very reassuring to us. And then furthermore, if we believe these to be real, we would hope that they would correlate with things that are known in the biology of autism spectrum disorder, right? So we took actually normative spatial transcriptomic data available from the Allen Human Brain Institute. So this is 6 individuals. We mapped it to that power parcellation I showed you earlier, which we were using to create our resting state functional connectivity matrices. And we ran a partial least squares analysis here, and trying to predict, in this case, the atypical connectivity within each subgroup that we had estimated relative to healthy controls, using the, again, normative Allen Human Brain Atlas spatial transcriptomics data to each of the brain regions, right, that have been used to generate that atypical functional connectivity. Does this make sense? It's a little complicated. The idea is, like, if there are spatial patterns in the distribution of genes that predict differences in atypical connectivity, this would be a kind of analysis or prediction that would be able to pull it out. And it does. Here's our first sanity check. We would hope that we would find enrichment of ASD genes in these individuals. We do. So there's strong in genes that are known to be downregulated across various libraries, including from Spark and SPARI, and one that we made a number of papers for ASD downregulated genes. These are downregulated. They have negative coefficients in the model. Published work that is upregulated has positive coefficients in the model. Again, that is more of a sanity check. Non-psychiatric gene sets for large part we do not see. So we do see positive with autoimmune and arthritic, which has been reported. This is known in the autism biological literature. Interestingly, again, I'm not going to go deep on this, but we do see subtype specific differences that map somewhat onto For example, remember subset4 is relatively spare. They have a lot of negative loadings across these genes, related in this case to other correlated disorders in psychiatry, and genes related to vocal learning, schizophrenia, which is known to have some correlations, ADHD, MDD, Tourette's, but not things like aphasia or disorder. And there's some interesting patterns across synaptic segments, she says. Again, the broad point here is that down a fairly long pipeline where one might be a little suspicious, but we are able to find reasonable, strong correlations between the biology, the spatial expression of proteins across the brain, and differences in atypical connectivity across these subtypes of ASD. Yeah.
How are you doing the pairings of genes to the variety of different behaviors that are shown here?
They're like where do these categories come from?
Yes.
Previous published work.
Okay.
Yeah, this is not, we don't invent these.
Yeah.
Yeah. Yeah. So these are like from SPARI, there's from the SPARI Spark Initiative. These are largely from one paper blanking on the first author of right now with some small modifications. So these exist though already.
Yes, okay, I just now am able to make a text at all and recognize that you're excited.
Sorry, yeah. Sorry. I should, yeah. It's a busy slide. I should add some more citations. I apologize for that. So just some additional controls because we wanted to really convince ourselves here. Again, we show you that abide versus endar. We kind of applying to totally separate data sets the same procedure. We get similar results. We tried a bunch of different clustering methods and selection criteria. So we use K-means with cosine distance, special clustering with cosine distance, hierarchical clustering Euclidean, Ward, and also with cosine distance and using average linkage, and these were all consistent. And you can see them supplement with the original results. We used 6 different model selection criteria for the clustering. They agreed. There's of course questions in this cohort because it's across the lifespan here in by age. So we restricted ourselves to a narrower age range. We looked just between 8 to 18, and the results are broadly consistent with the main analysis. We also confirmed the mixed effects model that age had no significant impact on the clinical behavioral features, or rather that the subgroups variability was not explained by age. We tried a number of different parcellations in gene analysis. So in addition, so we also used the Craddock parcellation in addition to the power outlets, and we, in addition to the human brain, Allen human brain atlas data, we used this brainstem gene expression. Again, the result you can see for yourself. The results are very similar, and then we Amanda had this very nice last text binding analysis. I'll walk you through very briefly. So we looked at the dimensional analysis, the PLS for each subgroup. We essentially then, of course, I think I walked you through this part. There's a number of things. How do I shorten this? We found the genes, ranked them by importance, identified their stability using bootstrapping. So we used shuffling and a heuristic P threshold to determine which were significant. Using the significant genes, we calculated protein-protein interaction network using strongly weighted genes, identified hub genes up to the 10 most connected genes, converted these to entry IDs and mesh IDs, and then downloaded the PubMed abstracts that included the hub genes from this analysis, and then counted the number of times each behavioral keyword appeared in the PubMed abstracts associated with the hub genes. So the idea here is to basically find, like, say, inner analysis, which are the genes that are really making a difference using PPI analysis, and then go back to the literature and say, is the relative frequency of each of these subgroups hub genes associated with the behavioral keywords we would think based off of the groups we showed earlier. So this is here just to remind you. And again, we're kind of seeing this large difference in group 4, where in particular you once again see the split, the verbal language social is kind of nicely recapitulated from real IQ in these subgroups 1 and 2, and 3 is demonstrating again significantly reduced actually worse. Sorry, in this direction it's worse, significantly worse restricted and repetitive behaviors. So again, this is it's a sanity check, and I think it's actually a nice prototype for a way to go back to literature and check genes that you think are important in these analyses that involve a lot of steps to get to, right? There's, in addition to the pre-processing, there's several subsequent analyses. So I think this is worth checking out if you're interested. And one last interesting thing, so Alessandro Rossi and Wenderoth Lab have recently found across 16 different autism mouse models. So there's some parallel results that there seem to actually be these subtypes. Again, this is rather far, but it's a nice kind of parallel bit of research, and the paper's very cool. So I want to.
Is it based on
Like-
genetic subtypes or is that gene subtypes?
Yeah.
Are there similar sort of genes inside?
There are, yeah, there's like shank mutations that are actually conserved. These are mostly conserved mutation models. It's nice. Yeah. So it's nice in that regard. Obviously, mice are not.
No,
Yeah, but the subtypes and behavior is interesting. Yeah.
It might be an unfair question, but do you know what the sex balance is in both mouse and human data sets?
It's not an unfair question at all. In the depression data, there's a bias toward women. In the autism data, it goes the other way. Yeah. And so I think it's 60-40 in the MDD data and like 70-30 in the autism data.
Would you be able to use any of the findings kind of as perhaps a talking point or leverage primer or something to argue for, I guess, a more updated version of these systems? I've used the ADOS
Yes.
before, it doesn't do an amazing job. Like, it's kind of a blunt instrument, and it was getting calibrated on mostly male participants. So.
Exactly. I think that's a terrific critique and one we're aware of. We went with we needed a certain number of participants to do the analysis, but you're right, it's very impoverished, and also it lacks a lot of symptoms that would be of interest, including, I mean, sensory, gut, like sleep, like all of these things that you think would be very relevant to measure weren't measured. I agree, it's a blunt instrument, but this was our first pass.
Yeah, the text was really nice.
Yeah, it's yeah. I mean, I think it's a very cool analysis. Great. Okay, so that gave us great hope that these subtypes might have some utility. However, not everyone really agreed with us. And so immediately after publication of Drysdale, this preprint appeared, evaluating evidence for biotypes of depression, attempted replication. Attempted replication is already a little dicey, so we're concerned. This was subsequently published, and in one of the main bullets of the paper describing its findings, where we argue that the methods used in Drysdale et al. Do not provide convincing evidence for biotypes of depression. So this might apply to us. And we decided to go back and see why the results of Dinga and Andrew Mark and me have differed from what we were finding, because we were concerned. The main thing, they did a nice analysis and replication of the methods, but the data was very, very different. And this is I think critical. This is a miss. Like, I worked on both sides of neuroscience and statistics, and often if you're in statistics, you kind of go with the data that's available, reasonably. I think it's also but it's important to remember, like, and this is getting better as I think this data it becomes increasingly public. At the time, there was a lot of I can't say too much here. There was the many. It was a different time for data sharing when we got this data set, and they got another data set. Again, the numbers per scanner, this is 2 scanners. They had 46 from 1. Like, that's not such a big difference. The diagnostic status is a huge difference. So here, in our study, all the subjects were actively depressed psychiatric outpatients. They were in the midst of a currently active MDD episode, which is important if you're studying depression. And they had moderate to severe total symptom scores. And so there's other details, but these were significantly depressed people in an active episode of MDD. This group were psychiatric outpatients who met DSM 4 criterion for one or more of the following: MDD, panic disorder, social phobia, generalized anxiety disorder, or with no specific requirements for active symptom severity or other exclusion criteria. So that's a significant difference. And I'll come back to, there's actually reasons to think more recently that it's possible maybe with MDD patients who are remitted, you may also be able to classify them as depressed. I'll come back to that later. But in this particular case, we'll show that this is too heterogeneous of a group, at least at this sample size, to really distinguish groups. That doesn't really matter as much. There are some differences in motion correction, but this is the real thing to pay attention to, the difference. And just to convince ourselves that we haven't done anything crazy, we used an analysis. This is a nice method from Bradley Efron that I want to apply for looking at many correlated variables and correcting for those correlations in order to do sort of an omnibus test of, in this case, bootstrap correlation coefficients between the symptoms. Here are our 17 Hamilton depression items and all the resting state functional correlations. So the idea here is simply what we would like to see is something poking over here showing that there are significant correlations between the symptoms and the brain, right? We do not see that in this data set. We do, however, see it in our data set. So this was the most baseline level sanity check to show that there are significant correlations in actively depressed individuals between the Hamilton and the resting state functional connectivity, but they're not in this more mixed population.
Your sample size was 40 something. Your sample size was 40
I think it was per scanner.
per scanner.
Yeah, and I think they had 4 scanners.
Okay,
Yeah, there was a mean N per scanner.
meat per scanner.
Yeah.
That's right.
So the sample size wasn't unreasonable. The problem was really, we think, from our analysis, the heterogeneity of the sample. I mean, you can imagine, like, I think it's a really interesting question, right? Like, you can imagine situations where you just had, this is increasingly common now, and I think exciting to think about. Like, maybe if you have 10,000 patients, you can start pulling out these different subtypes. But at the sample sizes of a couple hundred, it's not. And I'll talk kind of a little bit about that, why that is, I think, in a second. Again, and there's a growing evidence now in the literature for biological subtypes. We're feeling pretty good. There's this replication of the Drysdale work from Katie Dunlop at Toronto, and just accepted in biological psychiatry, showing in a totally different sample, this 3D data set, that the same or very similar subtypes hold. But, and this is a big but, I'm sure many of you are familiar with this paper. So last year, this paper came out suggesting that reproducible brain-wide association studies require thousands of individuals. So this is just saying cross-sectional work, like we're doing here in the subtyping work, in which we just have one measurement or a couple measurements per patient cross-sectionally, aren't going to work at a few hundred individuals, which all of these subjects are. And in fact, their claim is essentially that those findings that are at that sample size must be due to file drawer effect. It must just be that, occasionally we get a finding and that's what gets published and everything else is swept under the rug. It's due to variability. So their general claim here is studies the association between each individual variability and human brain function and cognitive behaviors or psychiatric symptomology. So it's very general. It certainly covers us as well as a lot of cognitive neuroscientists. And the main claim is that any brain-wide fMRI investigation studying correlations between brain and behavior across a population requires thousands of individuals to be reproducible. How did they come to this conclusion? And well, first, a big problem with this, certainly it's a threat, obviously, to our work where we're doing this, but in general, why would you fund or pursue fMRI population neuroimaging when it's $1,000 or a couple of very expensive per participant? And the effect sizes that they're reporting are, correlations of 0.02 for the Human Connectome Project ABCD and UK Biobank project. This is their figure 2. Furthermore, they're showing why would we believe past results using US analysis, including multivariate ones like ours. This is using a psychopath pathology measure in the CDCL data set. And they're showing that out of sample R's are close to 0. And I'll unpack this for you a bit in a second, but if thousands of individuals are necessary for reproducibility, and most previous work has tens to hundreds of individuals, previous work is not reproducible or believable. So based on their plots, we'd likely conclude that effects in cross-sectional fMRI studies are probably due to selection bias in publication. And this was not missed by media. So as you probably noticed, this led to a lot of speculation that fMRI maybe isn't a great place to put your government money. But again, we'll come back to the first thing here is the importance, as I'm sure any of you who do machine learning know, of nested, careful cross-validation. Because what it turns out, there was a very nice rejoinder that came out to American Terrible Clemens. They essentially used PCA for dimensional reduction, kept the top half of the principal components, and then used support vector regression method in this case, on the full discovery sample across a range of sample sizes, and then compared for each sample size the prediction on the in sample versus the out of sample, and that's what gets you this figure here. That's what they're showing you. Here's in sample, here's out of sample, and here's different sample sizes. So each of these dots is a different little experiment they ran on this large data set. So they take, in this case, 25 individuals. They fit their PCA, they fit their support vector regression. In sample, that particular run would suggest that they would have a correlation of 0.8, but then when they did it out of sample and held up data set, they have 0, right? Does this make sense, this sort of plot? And so in here, then they do a shuffle test and get some threshold of significance, and they're showing essentially that like at 200 on average, this is the red dot, things are still not significant. You really need 1,000 to make it significant, hence the claim you need thousands of individuals to sort of study. So, first issue, as we get past the animation. They do it like this: training, testing. There's an issue with that, and the issue is that what you really want to compare is what you think the in-sample generalization would be, not the overfitting of in-sample versus then the outer sample generalization. So they missed that point. There's a few other points too, but that was very easily immediately picked up by in this nice rejoinder by specifically Torweger, and they do the same thing, use PCA, fit in a grid of principal components using very explained, just like American Terrible Clemens. But critically, they do this training, validation, test, and out of sample. So now we can say, okay, we're gonna tune our hyperparameters, they're also tuning hyperparameters, tune our hyperparameters here, test, and then this is gonna be our in-sample estimate of how well it's generalized, and then out of sample, we'll do the same thing. We'll keep this holdout sample for that axis. And they do this on the HTTP data. They show it with kind of using the method without, they call it without cross-validation, but this is the Merrick, Terrible Clemens et al. Kind of result, which results in this massively inflated in-sample estimate, and then your out-of-sample is low. They show that just using correct nested cross-validation, they have what you see here. These are the averages for different numbers. You get the kind of nice line that you'd expect, where with increasing sample size, you're having accurate predictions of your in-sample and out-of-sample. Since we were working with ABCD data, we wanted to replicate this for ourselves, so we did the same analysis. Exactly, because what they had in their study was the ABCD data, so we replicated their analysis exactly, and what we find is, again, this is for the NIH toolbox, so kind of cognitive measures. If we look at groups going from 33, 80, 200, 460, and 1,964 on average, we see this, again, this very strong overfitting, as they showed. But if we do appropriate nested cross-validation, actually pretty small sample sizes, particularly for the neurocognitive effects, which are very, the cognitive behavioral effects, which are very strong, generalize pretty well. They obviously have more variability down here, but they generalize pretty well in the 10s. So that's a significant difference from their finding. We did this for both this cognitive ability measure, again, getting the sort of hockey puck overfitting effect from using their method, using a regularized canonical correlation analysis, getting this unbiased estimate, and for psychopathology. And psychopathology seems like it's more complicated. Maybe you need like about 100 individuals to reach significance. That's not surprising. Also remember from our lesson before, this is a pretty mixed cohort. A lot of these are actually healthy controls. So this is not the most well-powered thing to find psychopathology, but nonetheless, this is not thousands of individuals. And we can work this out across a full curve in various different ways. If you want to look at reproducibility, we can look at reproducibility of significance. That is, are the findings significant across these different replicates? We can look at reproducibility of effect size, and we can look at reproducibility of the coefficients. Are the coefficients that are estimated stable? All of these show this interesting saturation function that actually we're now working up is related to some nice historical findings in random matrix theory that these fall right out of. But for significance, the training set sizes, here we're looking at support vector regression or an RCCA. Support vector regression is dotted, RCCA is solid. For these particular data sets, for significance, the NIH toolbox cognitive behavior saturates very rapidly. You're fine probably in the tens of subjects. And then you maybe need on the order of 200 for psychopathology, 400 if you're doing an SPR. Interestingly, for effect size, we see the similar thing. These are kind of like the dot averages you can see before. So it's a nice pattern. What's interesting is that this is on a log scale, the particular data set sizes that they chose. So if you actually just plot it on the scale of the data here for the SVR NIH, the RCCA NIH, the SVR CDCL, and the RCCA CDCL data, you again see these interesting saturating curves. And these relate, if any of you are familiar with Ian Johnstone's work on PCA and admissibility of PCA in high dimensions and how that relates to the random vectors in high dimensions and how they eventually converge to their true underlying value. This is exactly the same phenomenon, and you can mathematically derive these curves and you can fit data, which we are working on. I think it's very interesting. That's an aside. This also is very apparent in the coefficients. So punchline, reproducibility of significance, effect size, and coefficient estimation don't benefit from thousands of individuals. Not only do you not need them, you probably are earning money past a certain point. So we found a very contrary result to theirs. Leading to a few conclusions here. One, reproducible brainwave association studies don't require or benefit from thousands of individuals, but they do require careful use of cross-validation. Tens to hundreds of subjects tend to yield reproducibly significant results consistent with previous findings in literature. And out-of-sample improvement is logarithmic in the number of samples. There's this particular interesting curve that characterizes in multivariate analyses the diminishing returns past a certain number of individuals. In this case, for the NIH toolbox, tens, and for CBCL psychopathology, low hundreds. And then from the previous, from the what I talked about just before that, cohort is very important. So diagnostic status, side effects, inclusion, exclusion criteria, all of these can have very important effects. It is in particular very important to track the diagnostic status and make sure that is sufficiently active to be used when you're studying. And then in large consortium study, effect sizes for non-normative effects may be significantly diminished by inclusion of many normative individuals. So that's, I might be really off on time here, but that was sort of part two, the controversy and some of our answers to it. And then I want to come last to a new approach that came really out of like discussing some of these issues and then also addressing like the most frequent question probably asked in early on, which was, are those really clusters? Like it looks kind of like maybe you just superimpose clusters on a pretty continuous thing. And very reasonable because fundamentally in doing the two-stage process of using a linear Gaussian assuming method like PCA and then imposing clustering on it, the first thing you're going to get out in that lower dimensional subspace is always kind of, is generally, it can be better separated, but frequently with fMRI data, it will look like this. And you'll be left scratching your head as to whether or not these are separated. Again, these are fit with kernel density estimators. In the ASD data, there was perhaps, again, in this different way of modeling the data, some consistency with different modes in the data that would lead you to think that the video would be maybe a little more convincing than clustering here. But we wanted to think of a better methodology that would be able to allow us to get at are these clusters The fundamental problems I kind of mentioned or hinted at was this two-stage embedding and clustering is suboptimal. And this has been known for a long time, since the 80s. Obviously, it's a algorithmically convenient way of doing things, but fundamentally, when you use something to embed and then something to cluster, the embedding method isn't optimized for the clustering method isn't optimized for the embedding. This can lead to issues. So it's just a toy problem. You can imagine this is our ground truth. If we have, this is in 200 dimensions. I'm just showing you the first two variables of 200 here. If we do spectral clustering followed by PCA, we do not get a particularly good clustering for accuracy on this known ground truth. If we do PCA, then spectral clustering, which is closer to what I've been discussing, it also fails. The method I'm about to present is sort of custom built for these types of problems, and so unsurprisingly, our joint data prevails. This is again Amanda Booch in the lab, who published this nice paper, which just accepted at AI Stats. So here's a particularly hard clustering problem. Again, we're in 200 dimensions with 100 observations. There's two classes. They are catastrophically overlapping. It's like an XOR clustering type problem. And it's very easy to see how this would break PCA and clustering. If you your principal components, it finds some directions of variability that are confusing because of the cluster structure. You cluster, it's garbage. Not really surprising. What we're going to try and do is solve this in a joint embedding and clustering penalty, where there's interesting, deep clustering can do this. I want to say like upfront, there are definitely neural networks that are good at joint clustering and embedding, but they usually take a lot of data. And what we're trying to do here is something that works in small or more sample limited cases that are high dimensional and something that is more explainable than those methods. And so the way we sort of built this was we're going to have some sort of, in a kind of empirical risk minimization framework here. I'm just giving you the optimization problem. Sorry, it's basics. Like we're going to minimize some embedding x hat. That's like our first three principal components. That's this PCA relative to the actual data. So we want it to be close to the data in some loss function. Say squared error. Yeah.
Just to orient myself in the context, we are thinking more so of, in the context of neuroscience, I guess, the functional connectivity aspects that you're looking to reduce and then cluster based on some behavior.
Exactly. In fact, where this will lead is first we're going to build a version of PCA essentially that clusters. Then we're going to build a version of CCA that clusters. Once we have a version of CCA that clusters, we're right back to the CCA problems that we had in the first part of the talk. Does that make sense?
Yeah.
Okay, good. Good question. So we have some data, we have n observations and p variables. P is much larger than n, and we want to find this x hat in a manifold. Again, say it's just the first three principal components that minimizes proximity to the data, but also is going to minimize this weird convex clustering penalty, which is the convexification of k-means. And essentially what this is going to do is we're going to look at our low rank representation, and for each row, we're going to say, try and minimize the distance between rows. So what this does is as you crank up this penalty, imagine you put this penalty at infinity, right? All the rows of x hat have to be the same. You have one cluster. Now you start relaxing lambda, it pops out, two clusters pop out, three pop out, and so on. So this is nice, it's also related to sum of norms clustering, some nice methods from control theory, and also the group lasso. If you're familiar with the group lasso, this is a similar thing. Because this distance is invariably something that looks like a not squared L2 norm or a group lasso penalty. So that's the setup. And if we apply to that problem I just showed you, there's two flavors here. Just briefly, one of them is penalized clustered matrix factorization is PCMF. You can ignore the other details. And this is a local linear version of that. So we were able to make it locally to get a nonlinear clustered PCA. We use something essentially like local linear embedding, but the clusters, that's on the right. And again, we start with a high lambda, and that'll give us one cluster, and we relax the lambda, and these do a good job of finding the clusters. Again, the true clusters underneath, and these are the estimates plotted at the top. We can show this a little more graphically here. What's nice is if we look at one of the coefficients for one of the variables in the model, we can plot its coefficient over time. And again, we're going to go from a very high lambda. We're going to relax lambda to 0, which is going to take us from one cluster to every point is a cluster. And over here, you can see again, first they sort of split and find the center of the means, and then they find the covariances. And that tends to be, there's this nice reliability to it, kind of consistent with PCA, where it's very sensitive to the means of clusters. Great at finding them, and then it sort of relaxes into these covariant structures. So that's the idea. It turns out that problem can be turned into this sort of wrapper function, and actually, I'm sorry, even better than that, just convex clustering. It can be used as a wrapper function in this. There's a lot here, but essentially all this is doing is iterating between fitting. This is an alternating method for fitting first for an x variable, then for a y variable. If you in the CCA problem, if your coefficients were a vector of a's and a vector of b's, you fix one and solve for b. You fix the other and solve for a. And that actually is not guaranteed to converge, but almost always does. There's more details on that. There's some nice work surrounding the alternating direction method of multipliers, which is sort of the backbone of this. That gives you some reasonable guarantees on that. But this gives us a nice way to do this pathwise clustered canonical correlation analysis, or P3CA, which we now have, as I showed you, both a linear and a local linear version of. So we can fit more nonlinear if we want. How does this do? We tried this out on a number of different public data sets. So a lot of gene expression data sets, some single cell RNA-seq data from mice, gene expression methylation, everyone loves MS fashion. So some images, some synthetic variable that we made to be very big. So with 100,000 observations, this would absolutely destroy convex clustering. It's not built to scale this well. So we, because of the 80 and MEM formulation that we used, this is one interesting detail, it naturally lends itself due to some nice work by Stephen Boyd's group in 2011 or so to this consensus formulation. We can essentially kind of mini batch it and then have an agreement, and it scales very nicely. And we tried it on multi-view versions of each of these. And you'll note that the sample sizes here can be range from very small to very large. And in some of these small cases, there's actually quite a few number of clusters. So those are hard on the other end of things. We just don't have enough data to estimate. And we compare this to a wide range. Our methods are in orange here, penalized cluster matrix factorization, the local lineal version of it, and then the CCA, the P3CA version of it. We compared it against basically what we've been using before, like PCA and k-means or PCA and hierarchical clustering, CCA and k-means. Ward spectral lactose subspace are more like Ward is more, of course, classic, spectral clustering classic. And then some very scalable methods like light in the vein, some more modern methods, Dirichlet process, Gaussian mixture models, this version of convex clustering, just to see how that compared, and then 3 deep neural network clustering methods that are relatively state of the art. These deep embedding and clustering, which do join embedding and clustering. It just so we wanted to compare the one method that actually tied us in the case, actually surprisingly this smaller data set, as well as the multi-view version of the same data set that we created by splitting the columns, is this deep embedding and clustering method. But by and large, our method did very well compared to a broad cross-section of methods you might use. We also applied this to some, again, much larger data sets. So here we have, this is just 400, but we have MNIST has 36,000, Dash MNIST has 36,000, synthetic data set has 100,000 observations. All of these, NA means you actually can't do this, so our method also has the benefit that you can fit a model on new data to do held out predictions. That's not the case of these other methods. And this X here just means, or the T means that things timed out, and the X meant they wouldn't run at all at these data sizes. So again, we're beating a lot of these other methods that are able to, in some cases, run on these data sets. And across the board. So it's a performance approach. We're very proud of it. And also, again, most importantly, what it's going to give us is something very different than this. This is the problem we encountered that I described before in a simplified form. So if we just look at the top selected rest 20 using our feature selection procedure, we just look at the top 20 selected resting state functional connectivity values from the Abide data set I showed you before, and we embed them, and we color code them by two of the clusters that we get by clustering in the first two components. We see immediately that the data has been projected down into this nice football that you kind of anticipate from PCA. If we run our method as lambda decreases, we get this very nice separation of clusters. And again, what we also really like about this is that it does connect the coefficients of this model, the linear version of it, not the local linear. That's more complicated, but still more explainable than for example, a joint embedding and clustering neural network. In particular, the linear versions of these come back with coefficients for each group. So you can imagine that if you have three clusters, it essentially is fitting three separate PCAs to those three separate groups, and you get the coefficients of those PCAs back. So you can interpret what's going on. Again, in the full autism data set now, the 299 patients looking at a larger segment of the resting state functional connectivity data. We actually, again, can trace out across different values of lambdas the coefficients. And here we're looking at the coefficients for social affect, restricted and repetitive behaviors, and verbal IQ. And so again, you get these for each coefficient, you get this interesting interpretability related to the trajectory and when these points divide. And as I showed you before, did I show this? I didn't show this. We have a wrapper that fits a nice dendrogram to each of these. So you get an interpretable dendrogram that's kind of telling you where things are splitting. That's in the paper. Also of interest, again, this is on the same data from the Abide data set. When we cluster, in this case, we're seeing pretty interesting separation. This is actually the worst RRB group out here, fairly separate. And we have these are sort of a more dimensional construct potentially. So this is again early days, unpublished, but we're excited to sort of now pursue like what is really a separate group that might have a more distinct mechanism, and what is on a spectrum of outcomes. And also somewhat of interest, there's a high reproducibility here between the clusters we got with the two-stage method, but it's not identical. So we're also pursuing that. I think that's interesting. Great. So I want to thank my wonderful lab again. Most of the work here was done by Amanda Booch. Some of the random matrix theory work that I touched on very briefly, she's working on with Preston Gibby, and a bunch of other great folks working on cool stuff. I want to thank Connor Lisson, who that first work, the MDD work is really out of his lab. I was working with him, and my collaborators and funding. My email's in the bottom corner if you would like to ask for papers and have any questions. Thanks.
Thanks, Logan. We'll take a few questions now, and those are going to retire to the other side of the campus to have further discussion if there's any questions here. I have two quick ones.
Okay,
awesome. One was actually on the algorithm I was noticing. So the lambda parameter gets tuned, and as that gets tuned, your clusters start to separate. So I think it's sort of a tuning parameter in some respects. It's a penalization term.
Mm-hmm.
But what is the stopping criteria for knowing when lambda is actually optimal for a given data set?
That's a great question. So there's for the sake of time, I didn't go into that, but there is. Do I have? I smart enough to include this. I'll come back to this. Check out this paper out. I'll just tell you because I'm not. This is currently acting in bizarre fashion. Okay. I'll just keep this up as a plug for this paper. There's really cool work actually starting here at Simon Fraser on this, and then at Stanford with Jonathan Taylor and Robert Tiptrani on this correlation statistic, initially used for selective inference in lasso problems. there was some nice work showing that that same statistic could be used to, as you're looking at the connected components of a correlation matrix, kind of determine how many is significant and when that stops being significant. And it turns out that sort of the dual of this problem is really has this, and this is nice work from Daniela Witten and one of her students on the theory side of convex clustering. It has exactly the same flavor, and it relates both to single linkage clustering and it relates to this correlation matrix thresholding or covariance matrix thresholding, and it related to our problem. And we plugged it in and we get like, it's a fantastic model selection criteria. So what's also interesting is in high dimensions, a lot of these other selection criteria can break. So if you really push the dimensionality up, they start failing. And in these problems where we were letting the dimensionality be big, this method, this correlation statistic was working quite well. So that's also in the supplement of the paper. But it gives us, we're actively pursuing that because we're excited about it as a high dimensional model selection criteria for high dimensional clustering problems. Yeah, but it's if you look, what it boils down to is essentially if you're coming along in your lambda path here, again, this is just for one coefficient, but let's say we're looking at the likelihood like function, like we're looking at sort of the joint, the value of the optimization problem of the data goodness of fit parameter plus the penalty. That like sort of there's points at which that sort of has a step function improvement when there's these cluster splits, right? Those can only happen at a point where the lambda has gotten low enough that suddenly it's worth splitting again. And those points create knots, and the distances, the inter-knot distance follows this correlation statistic. Developed here.
Sorry, like interdistance, that's distance in the change in lambda?
Yes, distance in like, if this is, it's, you know, this would be a big one, right?
Yeah.
Like, that's like way bigger than expected by chance. But next, maybe they start getting dinkier and dinkier and dinkier, and at some point you're like, the distance doesn't matter. And what's pretty cool is that there's a nice distribution for that, and you can see what would be a null model would be. We'll project that number.
Is he online? Oh.
Yeah. Well, in that case, yeah.
So to the extent that the clusters could be thought to be picking out biological subtypes, I wonder, I know you didn't necessarily want to go into it in person, but do you think they would be, the same clusters would be borne out by, say, oscillatory measures or other non-fMRI ways of looking at the brains of these people?
That is a terrific question. So we have, as part of the ongoing clinical trial, we're collecting EEG data on each of the 5 days during the same protocol. So as some of these people either go into remission or don't. And in the same patients, we have the fMRI data pre and post. We're very interested to see if there's some explainability between the two. But I don't know. I think that's an empirical question. And there's a lot of problems just with translating fMRI to EEG. Yeah.
I wonder if, say, the sample size that you would need to see the clusters would change. I mean, I don't know. I guess BOLD has a slower sampling rate by far than like a live EEG, right? Yeah. So I don't know how that would affect, or if it would affect, you know, because if what in the blood flow is important, what is signaling that, right? So maybe the BOLD signal isn't missing anything, and so you'd still need the same number of participants with long resting state EEG recordings to capture those events, even though the measures are a little different. I'm not sure I'm explaining myself very well.
No, but it's good. It actually touches on like an even slightly larger topic, which is like, there's like, we anticipate that something is changing in the physiology that's resulting in this response. But we don't want to assume that that what's allowing us to subtype people is the same as that thing. That is a terrific segue to this wonderful paper by Chuck Lynch that's currently on bioRxiv, where this is in Connell. He found they're looking longitudinally in patients who are going in and out of depression over time. And they're doing personalized kind of network analysis for these individuals. And what they found is a twofold expansion in just the real estate of the frontostriatal salience network in depressed patients. And it doesn't seem to change when they go in and out of depression. It's almost like it's either something like a scar from when they had their first episode, or it's just something that naturally predisposes them to depression, but it's more of a trait than a state. So there's this sort of question of like, even in that, and that could drive, if you imagine like the spatial distribution of these networks, like those are pushed across populations cross-sectionally. You could imagine that actually something like just this distribution could be driving the differences in correlations that are the representation that are then driving our differences in our algorithms. So it's not clear, I think at this point, whether or not it's a state, that there's something just about the brain organization or structure, or these, maybe these are defined by correlations as well, it's a little circular, but something that is a trait of the individual versus something that's actively changing. And of course, that's very important to try and understand, like, what is TMS doing, what's antidepressant doing? Because that separates the mechanism from the predictive method, if it's different. Yeah. That doesn't answer your question, but it's in the direction.
No, that's, I mean, my background is more EEG stuff,
Yeah.
so that's, very interesting to me.
I think, so I would look at this paper because it also seems, from the effect size of this, it seems like maybe it would be more likely. I think what our labs, and I think some people we've talked to are finding is that like maybe the dynamic changes in fMRI aren't as big as some of these other things, and so maybe EEG might be a better place to look for immediate changes that might be related to changes in synaptic formation and elimination or strength related to these interventions.
Right on.
Just noticed that the person that mentioned the UBCs on this paper with you.
Oh yeah. So there's okay. Good. Yes, it makes sense. It makes sense in the context. Okay, so I'll go talk to the doc.
Yeah, I've had the same thing happen where I've got authors of papers I've never read before.
I know. Yeah.
Great, thanks, Logan. Appreciate it.
Thank you. Good.